venture from the beginning onwards. Sorry for the disturbance. I thought the mic was on. Okay, listen here. This protein are embedded here, and the protein. These are proteins. The proteins which are embedded within the layer, that we are calling it as integral protein. That is primary. This one is labeled with this one integral protein, which are embedded completely in the layer. Wait a minute. Sorry for the inconvenience. I thought that the sound was on. Okay, I need to teach from the beginning again here. Okay, I will give a proper wind up here. That whatever I thought. Sorry for the inconvenience happened. See, cell membrane. What we are discussing today is of cell membrane of eukaryotic cell, and the cell membrane is composition will be different. The composition of the cell membrane. What it is made up of? The components of the cell membrane. See, yesterday we have discussed the cell chapter. In that prokaryotic cell is completed, and eukaryotic cell also we have started yesterday. So in eukaryotic cell, so eukaryotic cell means where do we see the eukaryotic cells? See, for example, plants, animals, and for example, protistans, fungi. All these are all these group of any organisms we will be seeing a eukaryotic cell. So you just take example of our own body. Our own body is made up of all the cells. It is of eukaryotic cells. Eukaryon is nothing but eu means true nucleus. Karyon means nucleus there. Eukaryotic cells these are. So in eukaryotic cells, it is made up of different compartments. So that in the last slide I said about that. So this when we see the eukaryotic cell here, so when we see the eukaryotic cell, so it is eukaryotic cell. U means true, karyon means nucleus. That is, true nucleus is there, and other compartments also seen. So when you are seeing any eukaryotic cells, it is you will be seeing the organelles there: nucleus, endoplasmic reticulum, Golgi apparatus, mitochondria, lysosomes, and centrosomes. Like that, you will be seeing cell organelles in case of eukaryotic cells. Whereas in case of prokaryotic cells, like in bacteria, this kind of compartments are not seen. So you might have seen compartments in case of train, how the compartments will be. So there are compartmentalization will be there. Like that, in case of eukaryotic cell, compartmentalization is seen. And upon this, what you are seeing, the white color layer, so the white color lines, these are cytoskeleton. Cyto means cell, skeleton means this. These proteins are there, no? That proteins are microtubule, intermediate filament, and microfilament. These are proteins there. So these proteins together to make a framework or a network-like structure within the cell in eukaryotic cells, it will be making a cytoskeleton there. So cytoskeleton is nothing but cyto means cell, cell skeleton it is. It will be made up of all these filaments, that is proteins it is made up of. And what we have seen is the locomotory structure in some case of in eukaryotic cell, like sperm, if you take flagella, that is the locomotory structure. Flagella will be there in case of sperm cell. That is the locomotory structure. Locomotory means showing the movement and displacing the position there from here to there. That is locomotory structure. And the paramecium, in case of paramecium, unicellular eukaryotic cell it is. Unicellular means single cell eukaryotic cell, that is paramecium. cilia is seen. These are locomotory structures. So, so far what we have discussed is eukaryotic cell and the compartments which are present. And even in yesterday's class, we have discussed about the difference between plant cell and animal cell. In plant cell and animal cell, what are the differences are there? The first one is cell wall. Cell wall is present in case of plant cell. In case of animal cell, cell wall is absent. And the centriole, it is absent in case of plant cell. In case of animal cell, it is present. And the central large vacuole, it is present in the plant cell. And in case of animal cell, it is absent. And one more thing, that is the plastids. It is present in plant cell, it is absent in animal cell. So that's what I discussed before that. And in case of cell membrane, so when we are discussing of the cell membrane, so we have taken one example of RBC there. So after discovery of electron microscope after 1950, the cell membrane structure has been studied. And especially when we are taking the RBC cell, so why do we are taking RBC cells in the sense? Because RBC cell membrane, it is easy to study where no other organelles are present there. Nucleus will not be seen in case of RBC cells. Nucleus is absent there. So due to that, RBC cells are taken for study. So that lipids, proteins and carbohydrates, and the composition is this. You can just take example of your house cells, your own home, where the walls is there, no? Walls are made up of what? Walls are made up of bricks, stones, cement, sand or soil, or some of the water it will be added, and the walls are constructed. Walls of our house or building. So like that, the cell membrane, so when we are talking about the cell membrane here, the cell membrane, it is made up of what? Some composition will be there, like in case of buildings we see, in case of house or constructed buildings, the walls of the buildings are composition of what? Let's say cement, sand or rock, bricks. So that is the composition of the walls of the building. So like that, if you see the composition of the cell membrane here, if you see the composition of the cell membrane, it is composed of lipids like proteins, carbohydrates and cholesterol. This composition varies from cell to cell there. So in case of RBC cells, the lipids percentage is 40%, proteins percentage is 52%, carbohydrate and cholesterol percentage is 9%. So this variations will be there. In some cells, the lipid composition will be more. Like you can take example of nerve cell. Nerve cell where myelinated sheath you will be seeing, no? That is made up of lipid composition will be high there. Protein composition will be less. Whereas in case of RBC cells, lipids is of 40%, proteins is of 52%, carbohydrate and cholesterol is of 9% there. And in case of this one, so when we study the cell membrane, so I am taking the cell membrane of this part. This is one cell membrane, even though it is a one cell membrane, but it has two sides, two layers will be there. So I said before that, see, if I am taking this as a cell and it is enclosed with the cell membrane. So this cell membrane, when I am taking this one, so this cell membrane, it is a one membrane only, but it has two sides. No, this is upper side, the outer layer, and this is inner layer. So the same one, this is outer layer and this is inner layer. The lipid it has outer layer and inner layer it has. Or if you just take a pen also, this is outer layer and this is inner layer. This is inner layer here. So in case of cell membrane, even though it is of one membrane, it has two layers, outer layer and it is inner layer. Inner layer in the sense what? It is exposed to the cytoplasm here. So inner layer, it is exposed to the cytoplasm here. This side cytoplasm will be there, and this side is outer side. This side is cytoplasm. This side is outer side. And this cell membrane bilipid layer, it is made up of lipid bilayer. These are lipids. And lipid has two ends. That is polar head and non-polar tail. Lipid has two ends there. Polar head, the round color one is polar head, and the this tail is there, no? This is non-polar tail it is. Non-polar and polar is with respect to the ionic charges and the molecules it differs there. So let me explain that here. See, I am taking a lipid layer, a lipid which is polar head. So this is polar head here and it is non-polar tail. So this is polar head. And this is non-polar tail. And this polar head is hydrophilic. This is polar head is there, no? This is hydrophilic, which is hydrophilic means water loving. Whereas non-polar tail is hydrophobic, which is hating the water there, hydrophobic. So if you see this layer here, if you see this layer, this is water loving area. That's why the polar heads are towards facing towards the water surface. This is plasma is there, no? This side it is cytoplasm is there. So that cytoplasm is nothing but where the liquid water surface will be there, and the polar heads are facing towards the watery surface, and the non-polar tails it will be present away from this water end there. And this is a lipid bilayer. It is a lipid composition. So the lipids are arranged like this. And how the proteins will be arranged? Proteins will be arranged like in Good Day biscuit. You might have seen a Good Day biscuit or Mom's Magic biscuit. If you see, this is yellow color one is that biscuit, and the red colors are there. No, that is cashew and badam will be there. So randomly it will be placed. No, so like that in case of cell membrane, the proteins are randomly arranged. The proteins are randomly arranged. So if I am taking this cell membrane here, so this is upper layer and this is inner layer, and the proteins are of two types you will be seeing. That proteins are of two types you will be seeing, that is this. So this protein which is embedded in the both the layers, this is outer layer and this is inner layer. The proteins which are embedded in both the layers, we call them as integral protein. Integral protein is nothing but the proteins which are embedded in the layers of this one. Either it can be completely embedded in the layer or partially like this, which is partially embedded. Partially in the sense it is completely embedded in both the layers, or it may embed it may buried partially like this. So partially embedded like this. So this is partially embedded and this is completely embedded, which are partially buried and completely buried in the both the layers of the lipid. So that we based on the ease of extraction. So based on the ease of extraction, easy, which one will be easier? See, a protein is present just like this upon this. This is a membrane and the protein is present just upon this membrane, and they are not buried in the cell membrane. And a protein is there which is present upon buried in the both the layers there. Which one is easier to extract, whether the first one or the second one? The first one is easier to extract, the second one is hard to extract. So based on ease of extraction, there are of two types of proteins we are seeing, integral protein and peripheral protein. So integral protein is one, so I numbered with this one here. So this is integral protein, and the second one which is easier to extract, so that is peripheral protein or extrinsic protein, which is easier to extract. So that is about the protein there. So lipid layer is this one. Initially the lipid layer was there, and upon this some of the proteins are arranged randomly, and those proteins is of two types based on ease of extraction. So the one is we call it as integral protein, which is buried in the both the layers. Either it can be buried completely or either the protein can be buried partially like this. In some proteins, those are we call it as integral protein, and in some the proteins are buried they are just present on the lipid layer that we call it as peripheral protein, which is easier to extract. And apart from what is there, say lipid we have completed and proteins we have seen here, and the proteins is of two types. One is completely embedded, these are of integral type, and this one is extrinsic, that is peripheral type. And next is carbohydrate, and these chains are there, no, the carbohydrate chains. Some carbohydrate chains, they are attached to this lipid layer here. So some carbohydrate chain, it is attached to this lipid layer, and some carbohydrate chain, it is attached to this um, the proteins here. So the lipid, the carbohydrate chain which is attached to this lipid, we call this as glycolipids. So glyco means what? Here carbohydrate, that is sugar, and the lipids is because it is attached to the lipids there. This we call it as glycolipid, and this one we call this as glycoproteins, because the carbohydrate which is attached to the protein, we call this as glycoproteins there. And this is a lipid bilayer, and it has a polar head. This is polar head, and this is a non-polar tail. These are non-polar tail. And carbohydrate is over. So carbohydrate chains are present outer surface of this layer, outer layer of this cell membrane, and some are attached to the lipids, we call it as a glycolipid, and some are attached to this proteins, we call them as glycoproteins. So glyco means carbohydrate which is attaching to the proteins, glycoprotein. So the glyco of this carbohydrate attached to the lipids, that is glycolipids here. So here this glycoproteins or glycolipids is present on this outer layer of the cell membrane, not in this side. Okay, so you need to remember that. So then how these will be helping? So you need to connect here. So this cell membrane, it will be having glycoprotein chains like this. The glycoprotein chain is there. It will be acting as a receptor, or receptors in the sense which receives the signals, or to send the signals is also there. To receive the signals in the sense to take the molecule. So if so and so positive receptor is there, in this first place the spherical molecule will come and sit. That is the another molecule which is coming and presenting there. So this glycoproteins are glycolipids, they will be acting as a receptor to send the signals. We can take simple example. So now my this part of leg part of, okay, hand, I'm this part of the body is getting ache or pain. So these cells got hurt, the cells got damaged. So now how the receiving and sending signals will be done? It is done through the neurons, or nerve cell, that is correct. But the cell which is required that particular chemical, now I am taking a painkiller, the painkiller has some chemicals there, so it will get dissolved in our body. That chemicals has to send to this part, this particular part. So how it will be sent? So now the chemical, it will be released in the blood vessel, and the blood will be taking that chemical there. So the blood circulates throughout the body, but the receptors, the pain killing that receptors, it will be present, the cells which are present here. And that how it will be framed or how it will be formed? It is just by like glycoproteins, in the form of glycoproteins or glycolipids. It will be having some receptors upon this. So they will be having some receptors here, and where the molecule of the chemical which is required to make or to repair this cell, it will come and bind here like this. So this is how the glycoproteins and glycolipids will be working, which will be present on them. And what about this cholesterol? There is another one is there, that is cholesterol. So that cholesterol will be present in between the cell membrane like this. So this is cholesterol. So the cholesterol somewhere you will be seeing like this. So what is the function of cholesterol? You might be thinking why so the cell membrane, how the cholesterol will be there. The cholesterol we say that it is bad. Why the cholesterol is there here means the cholesterol is there here for stability. So this is cholesterol which gives stability to the membrane. That we call this as cholesterol, which gives stability to the membrane. If the since the lipid layer it is made up of the cell membrane, it is made up of lipid layer. It will be fluidy. Fluidity will be there. Fluidity means which flows like this, which is jelly-like structure. So it will be like this. So this so that this membrane should not get disturbed. That it is made up of this cholesterol which gives proper stability to this membrane, that is cholesterol here. The cholesterol is the now the cell membrane composition is completed properly. So this part of the cell I am taking this membrane one fragment of this. It is containing two lipid layers. This yellow color are there no lipid bilayer it is which is made up of lipid bilayer, and in between that we will be seeing some proteins which are present in between that. So some proteins which are embedded in the both the layer are partially embedded or completely embedded. That kind of proteins we will be calling it as integral proteins, and peripheral proteins are nothing but which are present upon just a membrane layer. So that is which is easier to extract, that is peripheral protein, and cholesterol are there here in between this within this membrane which gives stability. That cholesterol is there. So it is the proper composition of plasma membrane, and this plasma membrane structure is there no the structure of plasma membrane, it was given by a scientist called Singer and Nicholson. So it was given by a scientist called Singer and Nicholson in the year of 1970. So that fluid mosaic model, what we call that is that was given by Singer and Nicholson. So in the next class, we will be seeing about this structure of now what we have studied is the structure of plasma membrane. So now in the next class, we will be seeing about the function of plasma membrane, how it will be helping in the functions we will be studying in the next class. So till that, take care and solve the questions which are related to this plasma membrane of eukaryotic cell. So you need to remember this is very important, this composition, and then especially these lipids are there. No, so when we talk about this lipid layer, mainly the major component of this lipid, it is made up of phospholipids or phosphoglycerides. So it is mainly made up of phospholipids there. So lipid which is not easier the normal lipid it is just a plus phospholipid it is. So it is made up of phospholipid, and the major one it is also called as phosphoglyceride also. In the last year also they asked the question related to that. Yes. The most abundant lipid in the cell membrane is butene, glycolipid, steroids, and phospholipid is given. So the answer will be correct here, that is phospholipid. Okay, now next is so the lipids are arranged within the membrane with. So the lipids are arranged within lipids. How the lipids are arranged like this? It was arranged. No, the polar head towards inner side and hydrophobic tail towards outer side. No, it is not. Both the head and tail towards outer side. No, how it will be? So head towards outer side and tail towards inner side. So we'll keep this. Next we'll read the next statement also. Both head and tail towards inner side not. So the head towards outer side and the tail towards inner side. The option C will be the correct answer. Okay. Integral cell membrane proteins are integral proteins which are see if it is a cell membrane, one is completely embedded like this. This is completely embedded cell integral protein, and some are partially embedded like this. Some are partially embedded. So completely embedded in the lipid layer, lipid bilayer, not. Partially embedded in the lipid layer, not. So lateral but not weight. So all of the above option is given here. So the option D will be the answer. Partially embedded also is there, completely embedded is also there, and it show lateral movement but not vertical movement in the sense. So it will this protein can move from here to here, but not from here to here or from here to here. So the lateral movement means from I am here. Now I am just moving this lateral movement. I am not shifting that to the other side. That is the lateral movement. So this both all these options will be accepted. So all of these are followed by this. So the most abundant thus we have completed now this. Plasma membrane of the plant and animal cell may not be seen in any microscope. It's not true, no? It is contain two layer of lipid and protein embedded within the lipid layer. That is also correct. So is not selectively permeable, not it is not, because it is a selectively permeable and can be only visualized with the aid of light microscope, no. So it is visible through the electron microscope. So the option B will be the correct answer. It contains two layer of lipids and proteins are embedded within them. So we have completed the just cell membrane of the structure of cell membrane. So cell membrane structure we have completed just now. The functions of cell membrane we will be discussing in the later. So solve any questions. If you are having any doubts, you can just comment. We'll solve that questions in the next session. Till that, take care. Thank you.